Okay, and welcome everybody to the second Evolutionary Medicine and Public Health seminar of this year. This week we are delighted to welcome Professor Stephen Stearns. Professor Stephen Stearns specialises in life history evolution, which links the field of ecology and evolutionary biology, and in evolutionary medicine. Steve is the author of seminal works, which I'm sure many of you watching will have read, including The Evolution of Life Histories and Evolutionary Medicine. He's received a number of awards, including an honorary doctorate from the Faculty of Mathematics and Natural Sciences at the University of Zurich. And probably one of my favorite that he's received is the Devan Medal for Distinction in undergraduate teaching at Yale University. Pretty cool that Yale recognizes excellent undergraduate teaching, which is so important. Currently, Steve is the Edward P. Bass Professor of Ecology and Evolutionary Biology at Yale University. And actually, as we were just saying, it's just under 2 years ago that we were able to host Steve in Oxford in person when the Anthropology and Zoology Department co-hosted Steve for a wonderful few days of talks, particularly the really amazing talk about the evolution of aging. So it is a pleasure to be able to welcome him online with us today. And Steve, I'm going to hand over to you.
Thank you very much. And thank you for the invitation. And thank everybody for being here. And when I was invited to do this, I thought that what I would try to do is use the fact that I'm currently working on the second edition of Evolutionary Medicine to communicate some things that I found striking early in the process. It's going to take probably all the rest of this year to work through and update all of the material in that book, but I knew going into it that there were some things that I had found really exciting, and that's what I'm going to be talking about today. So forgive me if I stumble a little bit. This is the plan of the talk. So I'm going to start off in the introduction with some comments on things that a lot of you probably will find rather familiar, but that I want to emphasize simply because in thinking about evolutionary medicine, I really have come to realize how important they are, and they provide some background. So the intro is going to be about trade-offs, mismatch, and large numbers. And I'll try to show you how those themes weave through the rest of the talk as I go along. Then I'll be talking about a striking case in which some very large-scale ideas, life history, evolution, immunobiology, endocrinology, and metabolism, got connected. Then I'm going to go into a bit of detail on one inspiring insight. There have been a lot, but and I just picked one because time is limited. And that has to do with how placental development affects the risk of metastasis. And then I'd like to close with a case in which evolutionary medicine is improving outcomes. And there I'm going to talk about phage therapy, where there have been some pretty dramatic advances in an approach which is now 105 years old. So that's the plan of the talk. So if we take the gene's eye view, which is a popular way of looking at evolution, particularly at Oxford, for the gene, the only thing that counts getting into the next generation, and health and happiness aren't important unless they aid reproduction. Now, I think that that's actually one of the deepest sources of vulnerabilities in our body. So it's pretty obvious, and if you've been reading evolutionary biology, it's familiar. But I really want to emphasize that if the genes really are in charge, that explains a lot of why we are vulnerable to illness. And of course, from the gene's eye view, if you're trying to design an organism, the basic question is, how much should I compromise performance in one environment to improve it in another? And here, the point I want to emphasize that if your gene's sitting in a body or in a cell, then those environments could be young versus old bodies. They could be different seasons. They could be different places. They could be different genomes, or they could be different sexes. And to a gene, those are all just different kinds of environments. So, you're sitting there down on the DNA sequence and you're looking out, And all of that stuff affects your probability of getting into the next generation. And the principle that guides your quote decision, in other words, the thing that's going to determine whether you increase in frequency or not, is basically the greater the benefit that you can get in terms of your ability to get into the next generation, the larger the cost you'll tolerate to achieve it. So if the benefit's as great as babies, the cost tolerated can be as large as death. Now, I'm going to use that idea, and I'm going to emphasize something that I've run into that I'd like to stay on message about, and that is what trade-offs look like from the point of view of a young scientist. For some PIs, unfortunately, the only thing that counts is grants and prizes, and grad students and postdocs are only important if they aid publications. So I'm just rephrasing the gene's eye view here. The greater the benefit, the larger the cost that will be tolerated. And if the benefit's as great as a Nobel Prize and a place in history, then the cost tolerated can be as large as exploitation and bullying. Now, there are a lot of excellent, gentle, supportive PIs, and they come in all genders and ethnicities, and a bunch of them are in the audience. But I just want folks to remain aware that when there's motivation and opportunity, and the PI thinks they can get away with it, stuff like this can happen. I've seen it in both medical research and evolutionary biology. Looking back on a career of about 50 years now, and it won't be going away. So I just want to highlight it, stay on message, and say it happens and we have to keep pushing back. Now, I'll go back into biology. Genes can only reflect what has happened in the past. They can't anticipate. And because of that, the organism is adapted to the past, so it can be mismatched to current environments. And mismatch implies selection. A mismatched population experiences selection with an intensity that's proportional to the degree of mismatch. We are mismatched to our post-industrial environment, and we are experiencing selection, and we can measure that selection. Now, there are basically two large differences or two large ways of thinking about mismatch. One is in time and one is in space. So if it's a mismatch in time, and there we're thinking about, say, our post-industrial population versus hunter-gatherers, that's affecting every individual in the population. But if there's a mismatch in space, which is something which is studied with immigrant populations, then it's affecting the different parts of that gene pool differently. They're not yet well adapted to the new country. The relatives they left behind could still be well adapted in that environment if it hasn't changed. And this is what leads to the analysis of conditions like sickle cell trait in the UK versus Africa, or autoimmune disease in the UK versus Bangladesh. Those are both mismatch in space issues. And it's a different setup. It's a different genetic setup, really, than is mismatch in time. Now, how long mismatch will endure depends basically on three things. And now I'm just going back to basics of evolutionary biology. The size of the population. A bigger population can generate more mutations, and it has more selective events, and those are births and deaths, and therefore it can adapt more rapidly. It's got more variation, and it's got more opportunity for selection. The intensity of selection, that has to do with the correlation of the trait with reproductive success. A population can be trivially or severely mismatched. How the trait's determined, and this is something which is a little bit newer than the two things that I just said. Polygenic traits respond more rapidly than traits determined by one or a few genes. So the lactose tolerance case, for example, that's just one or a few genes. There are other traits, however, that are influenced by many genes. And if you have a lot of genes that are intermediate frequency, they're in the middle of their frequency range, and that's the range of gene frequencies that can respond most quickly. So if you apply selection to a polygenic trait, it can respond surprisingly rapidly. And the UK Biobank is this giant database that has not only phenotypic traits, but a lot of genomic data in it. And a lot of people have been mining it to try to see signatures of selection, and they have reported that they can detect genetic responses to selection in the UK for myopia, age at first birth, and age at menopause that have all occurred within the last century. So that's quite a different view of how rapidly contemporary human populations could be responding to selection. And the third thing that I wanted to talk about is large numbers. And the reason I want to emphasize large numbers is that I have repeatedly run into cases where people are talking about things they might want to claim are adaptations, but they're talking about things that happen rarely. And as I'll unpack now for a minute or two, that doesn't work. So large numbers give us the precision of adaptations, and to communicate that, I've put up Reverend Paley, who, of course, talked a lot about the perfection of natural design, and the vertebrate eye is often mentioned in those conversations. They also give us the risk of cancer, so a couple of pictures of melanoma and breast cancer. Now, how precise can an adaptation become? Well, it really depends directly on how many selective events have shaped it in the past, and selective events are mutations, births, and deaths. So if you're talking about something that's been going on since the origin of life or the origin of the vertebrates or something like that, there have been billions of selective events. And you can think of a sculptor who's carving a figure out of a block of marble. A few hits of the chisel, they produce a very rough piece of stone, but if you give that person time, they'll have thousands of hits, and then they'll follow it up by rubbing with millions of grains of sandpaper, and they'll produce a masterpiece. The precision of an adaptation depends not only on the difference that a gene substitution makes to lifetime reproductive success, it also depends on how many times mutations shaping that trait have been fixed. So just to get that idea across, here's a comparison of a piece of marble that was not finished and a really lovely statue from the 6th century in Greece that shows what it could become. The same general principle applies to things like the design of our eyes or brains or kidneys or you name it, any of the wonderfully perfected organs that we have in our bodies. They've all been shaped by billions of selective events. However, large numbers also create the risk of cancer. And the risk of cancer is proportional to body size because as we grow from a single-celled zygote up to our full adult size, the cells in humans dividing to make up our body, they divide about 40 trillion times. In a nematode, it's about 1,000 times, and in a blue whale, it's about 10,000 trillion times. So for us, each time a nucleotide is going to be duplicated, the probability it mutates about 1 in a billion, about 10 to the minus 9th. But we have about 3.3 billion nucleotides in our genome, and that means that each of us carries about 3 new single nucleotide mutations with each cell division. Now, if you put in the 40 trillion cell divisions, that means we have each of us experienced about 120 trillion somatic mutations. It takes less than 10 specific mutations for a cell to become cancerous. By the way, many of these mutations aren't going to have too much to do with cancer. They're going to be in portions of the genome that are not expressed or are neutral or otherwise irrelevant. But still, 120 trillion is a lot. And the probability, for comparison, of winning a big payoff in the national lottery in the UK is about 1 in 100 million. So the point is, cancer is given enough tickets to win the lottery many times over in each human. And this means that ever since there have been metazoans that are multicellular, evolution has been producing defense mechanisms that make that multicellularity stable and possible. It's been working on the problem for about 2 billion years. And fortunately, we have those defense mechanisms in our body. So those were my preliminary comments. And now I'd just like to give you a brief roadmap to the rest of the talk. It has 3 mileposts in it, 3 sections. the first section is going to be how the physiological system is designed, and it's designed to in a way that creates reproductive success in the face of trade-offs. And this is an example of connecting ideas. The second section is on the connection between invasive placentas and metastatic cancer. And I picked this one as an inspiring insight because it really blows my mind that there's a deep trade-off that shows how the body is exposed to a risk caused by large numbers of cell divisions, and just how early in our lives the benefits of that trade-off are realized. The third example is about phage therapy as an example of improved outcomes. The design of that therapy relies on the evolutionary trade-offs that are faced by bacteria. And the appropriate phage is selected from a sample that numbers in the trillions. So that is a case in which two of the things that I've mentioned in the introductory remarks are going to be featured. So let's start with connecting ideas. And by the way, I'd just like to mention again that my reason for talking about connecting ideas and having inspiring insights and improving outcomes is that I think these are some of the features of evolutionary thinking in medicine that really help to make it a cohesive field and show how evolutionary thinking pays off. So how should physiology be coordinated to achieve reproductive success? Well, Andrew Wang, Harding Luan, and Ruslan Medjetov recently is 2019, published a paper in Science that used insights from life history evolution to connect endocrinology with immunobiology. Boy, I wish that kind of thing was around when I was an undergraduate 50 years ago. Seeing such large-scale connections between evolution and physiology and how organisms are designed, I think is really very motivational. And it also, I think you'll see, gives us a handle on lots of interesting research questions. So the connections that they made explain the mechanisms that mediate or show how mechanisms mediate growth, reproduction, and maintenance. And they break maintenance down into dormancy and immune defense. And they summarize their messages in 4 figures. And these are a little bit complicated, so I'm just going to highlight them. There's more info in these figures than one can really absorb sitting there listening to me talk. So I just want to try to pull out some of the large-scale features. The first point is that metabolism is divided into stuff that builds us up and stuff that breaks down. So anabolism is construction and catabolism is breaking things down. And each of these processes is mediated by mechanisms that take an environmental condition and sense what's going on and then send a signal and engage a program. So that's anabolism is on the top, catabolism is on the bottom of the figure, and we're working from the left to the right. So if you're well nourished and you're healthy, then you're supporting growth and reproduction and you're relying on anabolism. But if you're poorly nourished and you're sick, you're engaging maintenance programs. And there are 2 kinds of maintenance programs. Okay, there's defense, and that requires energy. And then there's dormancy. So you can either push back or you can shut down. So dormancy is induced by nutrient scarcity, and defense is induced by infection. So what you see here is a schematic layout of the different players in the body that are involved in those processes. And here is another way of looking at it. The central control for all of this is the hypothalamus. And so it's up there on top. It sits in the middle of the brain. It's about underneath if you go in about 3 or 4 inches from the top of your forehead towards the base of your brain, you'll hit the hypothalamus. And it is sending out signals that are regulating, and I now want your eye to fall down to the bottom of the screen for a moment, and you'll see growth, reproduction, and maintenance at the bottom of the screen. Those are all life history trade-offs. And they're being mediated by these hormonal axes that are running from the hypothalamus through the pituitary out to the periphery. So the growth axis is being mediated by growth hormone and insulin-like growth factor. The reproductive axis is mediated by things like gonadotropin releasing hormone, follicle stimulating hormone, that's FSH and LH in the middle, and those go down to the sex organs. And maintenance is being mediated by the hypothalamic pituitary adrenal axis, which is on the right, and that's corticotropin releasing hormone, adenocorticotrophic hormone, and that's affecting the adrenal glands and others. So the result is a system that has sensors and feedback loops and allocates energy to different functions depending on the information it's getting from the environment. And the point of these pictures is that people who are engaged with physiology and with the design of the organism are now able to see how that fits into the major trade-offs of life history evolution. Now, this one is a complicated slide, and I only really want to pull one point out of it, but let me go over it a little bit. Up in the top panel, you have dormancy. Oops, sorry. I see, I just discovered I can flip these slides by tapping on my trackpad. So the blue part of it in the upper panel has to do with stressors, sensors, signals, and life history programs that have to do with dormancy. The orangish panel on the bottom has to do with stressors, sensors, and signals that have to do with defense. And the point of this slide is to show you that inflammation is engaging both defense and dormancy programs. So the dormancy programs are induced by resource scarcity, the defense programs induced by infectious agents. They both are picked up by sensors. They then use mediators to activate either anabolism or catabolism. And this coordinates a very complicated response. But the idea is if you're infected and your body has to respond with its immune system, there's going to be a trade-off, and that trade-off is going to be mediated by sensors and signals. And the thing I want to pull out on the right is this little note down on the right-hand side. It says, note the granular detail. It accesses mechanisms, because you can see over here, you've got a list of organismal and cellular processes and particular molecules that are identified. And this is kind of a shopping list of research programs that takes a big problem of how to respond to an infection and breaks it down into a series of issues that individual labs can address. So this sort of integrative overview is actually a big generator of research programs. And a lot of them have to do here with the inflammatory response to infection, where these mediators are engaging dormancy-associated programs. And so while the immune system is ramping up its defense, other parts of the organism are shutting down to resist stress and protect tissue. Boy, I wish that physiology had been explained to me like that 50 years ago. Now, the host response is really divided into resistance and tolerance. So resistance is a defensive program, and tolerance is a dormancy program. Resistance is using anabolism, and tolerance is using catabolism. And this distinction is really quite important, because whether you resist or whether you tolerate depends upon the costs and benefits of each. And you can only really understand whether you should resist or tolerate if you have a very clear idea of trade-offs in your mind. So this is from an earlier paper that Ruslan was involved in, and it's really just laying out in a picture the idea that there is tolerance to the pathogen, but there's also tolerance to damage by the immune system. And one of the big costs of the immune system, of the immune response, is the inflammation that it causes and how that disrupts other processes in the body. And understanding why it is that this very beneficial defensive response also carries with it all of the costs of autoimmune disease and the pain and the swelling and the suffering of inflammation, the release of free radicals, the increase in the probability of a mutation that will cause cancer, all of those things are entering into a trade-off. And we have to go back to that idea that the bigger the benefit, the larger the cost will be that will be tolerated. So those are the kinds of issues that come up when the organism is being designed to balance resistance versus tolerance. And the point of this photo, just to re-emphasize that before we go on, is that the host can reduce fitness costs with tolerance mechanisms that are either tolerating immunopathology or the direct tissue damage that is being caused by pathogens. So why do I think this first of the three cases is important? It really creatively combines life history evolution with endocrinology and immunobiology at the mechanistic level with a lot of detail. It is a significant advance in the field of evolutionary physiology. It complements and extends the older field of comparative physiology, which is what I was trained in long ago. And in doing that, it appeals to the interests and the abilities of a pretty broad community of biologists. It helps to recruit them to both life history evolution and evolutionary medicine. And it does so with the most seductive mechanism, which is neat questions and really intriguing problems. So it helps to unify parts of biology that had been fragmented, and it sends them into a future that's shaped by big questions. So I think that that is one of the real strengths of evolutionary medicine, is that it is helping to bring together population level thinking with organismal level thinking, and it's doing so in a system which is the focus of a lot of attention because of the difference it makes to the lives of humans. Now, the second of the three things I want to talk about is an inspiring insight. Now, there are a lot of them out there, and this is just one of them, but I think this one is particularly neat. And it started with a conversation I had with Betsy Uhl. Betsy is a veterinary pathologist at the University of Georgia. And in a side conversation at a meeting, she told me that cows and horses, which have non-invasive placentas, don't get as much metastatic cancer as dogs, cats, and humans. And she thought that was striking. And I agreed. And at the time, Alaric de Souza was taking a tutorial with me in evolutionary medicine. And so he made it the theme of his undergraduate senior thesis, which he handed in 2012, so 10 years ago. And he did that thesis with Gunther Wagner and Gunther's group. And Gunther was convinced by the data that Alaric assembled, and it led him to take a real hard look at that association. So a bit of refresher on kinds of placentas. On the left, we have sort of a basic placenta. Baby is on the left, mom is on the right. And you can see, if you work through going from left to right, that there are 6 cell layers. There are 3 each in the fetus and 3 in the mother. And they can all potentially contribute to the formation of the placenta. There are fetal endothelial cells, fetal connective tissue, and chorionic epithelial cells in the fetus. And then there are endometrial epithelial cells, maternal connective tissue, and maternal endothelial cells. The endothelial cells are basically surrounding the arterioles or the capillaries. Now, if we look over on the right, we see 3 different ways this basic setup can be organized. The one on the top with the 6 cell layers is called epithelial chorial, and that's what cows, pigs, and horses have, ungulates. The middle one is endothelial chorial. It has 5 layers, and you can see that one of the layers that's been taken away was taken away by the mother, that green layer. That's the one that went out. And what we have and what we share with rodents is hemochorial. And in the hemochorial system, the mother's blood vessels have opened up and they are actually pumping blood right past fetal tissue. So you can see that on the right, you see kind of a semicircle, and that's the wall of the mother's blood vessel with some blood cells in it. And the fetal tissue, which is the vertical blue bar, is actually bathed in maternal blood. So that's the kind of placenta that we have. And if you look at the phylogenetic distribution of placental types across the mammals, the important point is that our type of placenta is ancestral. This is what things started out looking like in roughly mid-Cretaceous time, something like that, about 80 million years ago. And I've put some red arrows here to highlight where humans sit and where cows sit, because you're going to see some human-cow comparisons. And it shows that the black part of the phylogeny is epitheliochorial, that's 6 layers, and the blue part of the phylogeny is hemochorial, that's 3 layers. So here is a slide from Alaric's senior thesis, and it shows that, in fact, cows and horses really do have less metastatic cancer than dogs and cats. All of these species have similar high rates of non-glandular epithelial metastases. That's the lower left of the 4 panels. But for the other 3 types, skin cancer, glandular epithelial cancer, connective tissue cancer, you can see that cows and horses have distinctly less risk of metastases than do dogs and cats. Now, primates have even more invasive placentation than carnivores. So far, it's kind of sounded like we're building up a story that says, hey, cancer's linked to placentation. However, it's not that simple, and that's because we can see, when we look comparatively with an evolutionary eye view, that the mechanisms that cells use to metastasize, to gastrulate, to move out of blood vessels, those basic functions are all present in marsupials, which don't have placentas. However, they are all shared with the way cells move into the endometrium, and that's called trophoblast invasion. So when the trophoblast, which is a developmental stage after the zygotes formed and started to divide, when it's embedding into the endometrium, the mechanisms that those cells are using to move into the endometrium and start to form placental tissue are all present in marsupials. That means that cancer cells are using mechanisms that evolved earlier than placental invasion. And therefore, placental invasion itself can't be responsible for the origin of malignant cancer. However, the invasiveness of the placenta continued to evolve after it originated, and it reverted to a non-invasive phenotype in ungulates, and it evolved even higher invasiveness in the great apes, including us. And that led Gunter and his group to refine the statement of the hypothesis, and they call the hypothesis evolved levels of invasibility. The paper, which is Kashitit's, it's 2019 Science, suggests that evolved changes in the permissiveness or resistance of uterine stromal cells to placental invasion are mechanistically linked to vulnerability to cancer malignancy. So as that quantitative relationship goes up and down in terms of how permissive or resistant the uterine cells are, so does the risk of malignant cancer. So when you look at it that way, the evolved changes are in the maternal, not in the fetal tissue. They're in the stromal cells that form the connective tissue of the endometrium, the placenta, and the uterus. And it suggests that the linkage isn't simply a case of antagonistic pleiotropy with the early benefit linked to a later risk of cancer. That effect does exist, but the story is more complicated than that. And basically, the story is more complicated than that because there's a lot of cell mechanism that gets changed. And it's not just an early genetic effect causing a late life risk. It is, a lot of this is mediated by fetal maternal conversation and conflict. So to test this idea, they did something clever, which is now enabled by many of the modern techniques of both cell biology and genomic science. They grew lawns of stromal cells from cows and humans, and then they watched the invasion of trophoblast cells onto those lawns of stromal cells from cows and humans. And they also measured, and this is where you get into really precise genetic technology, they measured which genes were up or down regulated in the trophoblasts and in the stromal cells. So in baby and in mom. This is what it looks like. You grow stromal cells on a grid. You measure invasion under the microscope, and you replicate it and do the stats. So this is one of their early setups in the process. You can see the grids on the left. In the middle, you can see human cells growing on human stroma and cow cells growing on cow stroma. And you can see the progress of an invasive front across the lawn of stromal tissue. And on the right, you can see the result, which is basically that the extent of invasion is much greater in humans, which is the blue bar, than it is in cows. So that was sort of the starting point where they could make their comparisons. And going on from that, they did a lot of further work. They showed that cow stroma resists invasion by malignant cancer cells, and that human stroma does not resist malignant cancer cells nearly as much. Then, by measuring the gene expression patterns, they showed that when invaded by trophoblasts, the stromal cells in humans and cows express different genes. Then they knocked down those target genes in human stroma and showed that if they knocked down those genes that differ in their expression in humans and cows, that will increase their resistance to invasion by melanoma cells. And basically what this means is that humans evolved greater expression of 6 genes that enhance invasibility, and that probably happened in the primate lineage. The conclusion is that the embryo invades, but mom lets it in. And by this sort of analysis, and I can't really do justice to it in the time available, they show that studying the genetic basis for evolved resistance to invasion reveals how stromal cells control cancer progression and identify therapeutic targets. So this is neat stuff, and I think it's really important. And it's important both at a practical and a conceptual level. It creatively combines the comparative method, cows and horses versus primates and humans, with powerful technologies that suggest potential therapies. Okay, so from a practical medical point of view, that's really neat. From a life history point of view, or evolution of aging point of view, it reveals a trade-off between performance extremely early in life with cancer risk much later in life. I mean, in our lives, in our life cycle as mammals, it doesn't get much earlier than landing on the endometrium and starting to penetrate maternal tissue to create the placenta. There are a few cell divisions before that, but not many. And it supports, for this kind of claim, for this kind of question, Gunter's claim, he sent in an email, nothing in evolution makes sense except in the light of mechanism. And of course, you're all familiar with Dobzhansky's statement that nothing in biology makes sense in the light of evolution. And Gunter was a bit frustrated at how frequently that had been quoted. But I think that both this research program that I've just summarized and the next one that I'm about to show really drive home this point that one of the really powerful ways that we can now do research and address very interesting questions is by combining the evolutionary perspective with the mechanistic perspective. And that gets me to improved therapies, which use selection by phage to reduce bacterial resistance to antibiotics. And in this case, The team that I've concentrated on, there are a number of teams around the world that are doing very good stuff with phage therapy, but the one that I know best is the one that was in my department at Yale, from which I recently retired. And it is led by Paul Turner, and Ben Shan really has been taking the lead on the phage therapy part of it. And I put a hero on the right, Ali Kodagaust. He was their first patient, and you will see a bit more about him in a minute. Now, why is phage therapy important? Well, here's something from the BBC last month, not quite a month ago. And they're reporting on an article in The Lancet that had just come out that showed that more than 1.2 million people died worldwide from infections caused by bacteria that are resistant to antibiotics. And a lot of that was lower respiratory tract deaths. There were 6 leading pathogens that were responsible for nearly 1 million of the deaths. And there's one particular combination, which is methicillin-resistant Staph aureus, or MRSA, that caused more than 100,000 deaths. And there are some other combinations. So the point is that there's a tremendous amount of antibiotic resistance circulating in the bacterial gene pool, and it is seriously disrupting medical practice. So the background on that is basically that multiply drug-resistant strains are now present in most hospitals, particularly in ICUs, where they're used a lot, and in many communities where they're a real threat in adult care facilities. And every time we come up with a new antibiotic, it only takes a year or two for resistance to evolve and to spread across the globe. And that goes back to the huge populations of bacteria and the fact that antibiotics were extremely strong selection pressure acting on them. So this has focused a lot of attention on how to make an antimicrobial therapy that is evolution proof. So here we would like to actually stop evolution in its tracks. One option is phage therapy, and that uses viruses that attack bacteria but not eukaryotic cells. So these are viruses that it is safe to put in our bodies because the viruses are engineered to deal with prokaryotic bacteria rather than with our cells. Interestingly, if you go back in the history of science, and Bill Summers wrote a very nice paper on this, it was a gentleman at the Pasteur Institute named Derel who discovered phage, which he couldn't visualize, but which he could determine were there by using advanced filtration techniques. And he did that in 1917 in Paris. And almost immediately, he used phage therapy. Between 1917 and 1920, he'd already made a whole bunch of attempts to cure bacterial infections using viruses. He actually came to the States, and he was a professor in the Yale Medical School for a while. Then things weren't working out too well. The community wasn't really ready for this radical idea. And it was Derel who went to Georgia and helped the Georgians to found the Institute of Phage Therapy in Georgia, which still exists. And that's the reason that Eastern Europe has had more of a tradition of phage therapy than the West. The West has just recently become, well, just recently, in the last 20 years, medical researchers in the West have become much more interested in phage therapy because of the problem with antibiotic resistance. So just to remind you, we're talking about lytic phage. They're viruses that kill bacteria, and they need to attach to a specific piece of bacterium. And when they get into it, basically they take over the machinery inside the bacterial cell to synthesize viral genomes and structural proteins. Those get assembled, and then phage progeny are released in a big burst. And so coming out of one bacterium, you can have thousands of phage. This is actually a very neat kind of therapy because it scales the dose of the antimicrobial to the number of bacteria that are around. The more bacteria there are to, quote, feed on, the more phage there are going to be. So it's a dynamic and response. It's not like you have to keep taking a pill. Now, the neat part of the idea that Paul and Ben and their colleagues had was that they figured out how to make phage therapy evolution-proof. And their insight, which is really quite clever, exploits a trade-off that traps bacteria in an unavoidable dilemma. So they go out and they find a phage that attacks the mechanism that the bacterium uses to resist antibiotics. They then let the bacterium evolve resistance to the phage. They said, go ahead, you guys can evolve all you want, and you can in fact evolve against the first stage of the therapy that we're giving you. But that causes the bacterial population to lose resistance to the antibiotic. So they then come back and apply the antibiotic, and that clears the infection. So the point is that the bacterium can either resist the phage or it can pump out the antibiotic, but it can't do both. And that's because it's using the same mechanism. And the trick is to find that mechanism and find the phage that attacks it. Now, this approach can also be used to guide the evolution of bacterial virulence. So if you know the part of the bacterium that is responsible for determining virulence, you can use a phage to attack that. They express that in this paper, which Courtwright actually was a senior author on. And they point out that you can either select a phage that will be dealing with the trade-off that has to do with antibiotics, that's on the left on the figure on the right, or with virulence. And the long triangles here are indicating the trade-offs. So if we look at antibiotic resistance, the particular mechanism that they attack is the efflux pump. So bacteria have a pump in their membrane that they use to get rid of poisons inside of them, and they're getting rid of antibiotics using that pump. And those receptors are signaled here with these little red plugs that are sticking out of the bacterial wall, and the rest of the efflux pump is transmembrane. It goes into the bacterium and pumps stuff out. And those are the ones where the phage-resistant bacterial mutants become more sensitive to antibiotics. Then a structural resistance factor would be something like a capsular antigen, okay? And that is what you can select with a phage that will modify virulence, and those would be the purple parts. So both of these approaches are basically conceptualized as exploitations of trade-offs that bacteria cannot avoid. Now, their first patient, that gentleman whose portrait I showed you, and it has appeared in newspapers, so it's not really a breach of confidentiality. He had had an aortic graft that had been infected with multiply drug-resistant Pseudomonas aeruginosa. So you can see a photograph showing the infection. This blue arrow right here is pointing to the infection on his myocardium. That was taken during an operation they had to open him up to try to get rid of the infection because antibiotics weren't working anymore. This is a CT image, which is showing the aortic graft and the collection site where they sucked some of the bacteria out. At the time, he was 80 years old. He's actually he was a very prominent ophthalmologist in Iran. He had spent 4 years fighting the infection, and he understood perfectly well that he didn't have any more options with antibiotics, so he consented to phage therapy. They used therapy just with a strategy that I've just described. It cleared the infection, and it didn't return. He did die 2 years later at the age of 82, but of other causes. So here's how they found the phage. They went out to a pond in Connecticut, and they took a liter of water out of it. It had tens of thousands of types and trillions of individual phages. They screened them, and they found one, which they labeled OMK01, that attacks the efflux pump in Pseudomonas aeruginosa. And there are automated methods now that you can use for screening, where you can take a population that's got millions of different things in it, and you can find the one that does one specific thing. They then confirmed in vitro that selection that's imposed by this phage causes Pseudomonas to lose resistance to antibiotics. So that's the development pathway that you use for this kind of phage therapy. Here is a review paper, and it's got a lot of stuff in it. You're not going to be able to read it. So the only thing I really want you to notice about it is that by 2019, there had been published reports, 5 published reports of cases in which people had been cured by phage therapy that were done in Western labs. This has been going on for quite a while in Russia and Georgia and Eastern Europe. The clinical trials had mixed results. And in fact, Ben and Paul currently have a clinical trial going on, and they're still recruiting patients into it. So what we have at this point in terms of evidence is very plausible case studies, but not yet population level clinical trials. However, that hasn't stopped them from saving limbs and saving lives. So Ben has used phage therapy to save the limbs of 5 patients who had undergone repeated surgeries and had developed multiply drug-resistant bacterial infections. The phage were either applied directly during surgery or intravenously, and the bacteria that were being attacked by phage included staph and Klebsiella. He's also used phage administered as inhalants, so through the nose, to prolong the lives of more than 20 patients that suffer from cystic fibrosis and who had developed infections with multiply drug-resistant Pseudomonas aeruginosa. After being treated with phage, the concentration of bacteria in their sputum went down by more than 3 orders of magnitude. Their pulmonary function improved significantly. So this is the kind of therapy that's now in the stage 1 clinical trial. And there are about 5 to 10 other teams in North America and Europe that report similar success. I can see I still have a few typos in my slides. I went over them quite a few times. Sorry about that. I want to give you some details on one such case just so you can appreciate the potential to relieve suffering. So there was an immunosuppressed elderly woman with a history of brain aneurysms who had vertebral osteomyelitis complicated by metal that couldn't be removed, that had been installed with a posterior spinal fusion, which you can see that kind of operation. This isn't her, but this is that kind of operation is done on the spine of elderly patients to stabilize the spine. Her wound actually burst open. It exposed hardware with deep tissue cultures that were growing Pseudomonas. She went back in. She was opened up again and drained and treated with 2 antibiotics, but she couldn't be treated with fluoroquinolone, which might have worked because that would have risked aneurysm rupture. I hope you're getting an impression of just how miserable an old person in the hospital can be. If she hadn't had phage therapy, she'd have had to stay on IV antibiotics until resistance evolved, treatment failed, and she died. On IV antibiotics, she was suffering worsening fatigue. Her wound continued to drain. She was dehydrated by orthostatic hypotension. That means that just because she'd been lying down and she hadn't been able to move around, her blood pressure was dropping, and that was messing up her kidneys. She was dependent on transfusions. She had anemia of chronic disease. She had poor appetite. She had repeated flares of rheumatoid arthritis. Ben gave her phage therapy. He did it with IV by instilling phage into her bladder and into the spinal hardware drain. It completely cured her bacterial infection, and it greatly improved the quality of her remaining life. Now, if you have all of that stuff going on, clearly you're not going to be living too long, but at least she was rescued from much of the suffering that she otherwise would have had to endure. So I think the take home on this is that phage therapy is one really very promising solution to the evolution of antibiotic resistance. And it's going to help us to preserve surgery as we have come to know it in the golden age of antibiotics. And I think all of us who face going into surgery should be very grateful that methods like this are being developed. So I think this is important because it shows that phage therapy that's informed by evolutionary thinking saves lives and limbs that would otherwise have been lost. That's the kind of evidence that makes any doctor sit up and take notice and gives us compelling arguments for adopting evolutionary thinking in appropriate parts of medical research and practice. And I'd just like to mention that evolutionary medicine is now into its third decade. It emerged as a focus of interest in the 1990s. It started with the book by Randy Nesse and George Williams and the papers that they wrote. It's had some success. The journal that was founded in 2011, Evolution, Medicine, and Public Health, now has an impact factor of 5.25. And you can compare that, it's roughly in the same ballpark as Nature, Ecology, and Evolution, which ain't bad. Evolutionary thinking is now an organizing paradigm for research on cancer, infectious disease, reproductive medicine, and immunobiology. Almost every paper that you now read in Nature or Science or Cell that has to do with cancer has a statement in it about cancer as an evolutionary process. And they're using evolutionary thinking to try to understand how cancers grow and differentiate. I think both in the case of invasive placentas and in the case of phage therapy, we've seen that evolutionary medicine gets some of its most powerful insights when evolutionary and mechanistic thinking are combined. That's a powerful combination. And some of those insights are saving lives. So that's it. Thank you very much. Gabriella, you're muted, so I'm happy to take questions. And I think that Gabriella is the one who's been seeing the questions coming in.
Thank you for such an interesting talk. We've got a great presence on YouTube and a bunch of talks that started to come in during the presentation. So I'm going to start with the earliest ones and kind of make my way forward. And at some point, I'm going to interject my own. So the first one is by Yasir, and I'm just going to pop it up so you should be able to see it as well on your screen. Is age at first birth a trait that can be considered determined genetically?
Well, like any other phenotypic trait, it's the product of a genotype by environment interaction. So it is determined in part genetically, but in fact, it has an adaptive reaction norm so that organisms that are growing faster usually mature earlier, and those that are growing slower usually mature later. The genetic determination is involved in shaping the entire reaction norm, the entire plastic response to the environment. So the answer is yes, it's determined genetically, but yes, it's determined by the environment.
Brilliant, thank you. The next question is, it's split into two, but I'll just summarize the two together. By Anoa. Thank you for your talk, Professor Stearns. I would love to hear your opinion about the effects of stress during pregnancy, what they could have on the later development of a child. Professor Jazianska mentioned last week that environmental struggles in mothers in their last trimester of their pregnancy compromise the ovarian function of their children, for example. So the effects of stress during pregnancy.
One of the things that I've run into in reviewing the literature for the second edition of the book is a lot of very recent information that's coming out on epigenetics and on the way that genes are imprinted at different stages. And we already knew from the analyses of the Dutch hunger winter and the Great Leap Forward that events that occur in pregnancy shape the physiology of the offspring in ways that depend upon the period of pregnancy during which the mother was stressed. So what Grazina basically is doing is deepening our understanding of that important interaction. In humans, it's, of course, rather invasive to try to go in and look at the expression patterns of the genes in ovaries, especially if you think about trying to do that during a pregnancy. But I think that paradigm is one that is very likely to bear fruit, and that we are going to find that there are a lot of transgenerational effects of environmental stressors, and that they're mediated by epigenetic mechanisms. I've seen enough in my literature review to know that this has just got to happen, but we don't, in many cases, yet know the mechanistic details.
Thank you. Yeah, interesting question. I guess also at some stage we're going to have to start thinking about, is there a threshold whereby you can reverse or kind of change the course of the impact of stress on the maternal body during pregnancy, and what that might give to the outcomes of the offspring?
Yeah, if I could just go on a little bit about that. One of the things that we repeatedly discussed when I was teaching evolutionary medicine was gene therapy and somatic versus germline therapy. And if it is somatic therapy, then when is it safe to do it and when is it safe not to do it? And if you're doing somatic therapy that is intervening in a reproductive trade-off, then you want to think very carefully about whether or not the person to whom you're giving that therapy is likely to be reproducing after you give it to them, or has already done with reproduction. And in this case, where it looks like there's been some impediment to ovarian function, it sounds to me like if you could do somatic genetic therapy that would actually reprogram the epigenetic patterns of expression in the ovaries, that you might want to do it before that child starts to reproduce. But boy, am I worried about the huge black box of unknown trade-offs and the enormous potential for unpleasant surprises. So I think that this is certainly the kind of thing that we just have to be grateful that we have policies in place that say you have to have stage 1 clinical trials. You have to make sure that whatever therapy you're proposing isn't doing more harm than good. Because although there's great potential for good, there's also great potential for harm. Remember, we're talking about stuff which is right at the center of what genes want to do, which is to get into the next generation, and where they are most willing to tolerate very high costs.
Yeah, definitely an important warning. Okay, Emily says, do you think that food insecurity and its associated psychosocial stress in children could alter their investment in longevity and subsequent health outcomes?
Well, I think food insecurity certainly does. And I think the question is whether you can demonstrate that whatever else is being done by psychosocial stress is a significant addition to the direct physiological compromise that they experience because they've been starved. And of course, that's an extremely interesting thing, and it's the topic of a lot of investigation. I think that it is interesting enough and that there's enough preliminary evidence to certainly justify a lot of further research on it. But from what I've seen, it's difficult to conclude at this point that it's the psychosocial stress rather than the direct physiological impact of starvation that is actually triggering the subsequent issues.
Thank you. And also, I can see people's questions coming in, and if anyone has asked a question that Steve has already answered and they would like to add to their question or make or kind of further add to it, then please just comment. I know it's not quite the same as being in an audience where you might be able to go back, so I'll try and go back to you if you have anything else to add. Okay, Rose Stevens says, thank you for a wonderful talk. You say that the next most fruitful area of research for understanding life history trade-offs is to combine mechanism and evolution. That's part 1. Could you talk a bit about how we can investigate trade-off physiology in humans? How do you look at physiology out of the lab, account for environments, and conduct ethical experiments? Basically, any research studies you think we really need to be conducting, or questions ripe for answering. Great question.
Fortunately, we have in place all of the human subject committees at all of the universities around the world that are better poised to answer that question than I am. When I think about that, I think about people who are, say, intervening in the field in hunter-gatherer populations where they have consent and they are taking blood samples and they're using sort of natural experiments to try to contrast different physiological states and look at downstream consequences of that sort of thing. Grazina Yazienska, who talked last week, comes out of a research tradition. Peter Ellison is one of the people who has really pushed it, that looks at such interventions. And there are some things that can be done that I think are not too controversial. For example, you could ask a bunch of weightlifters to sign off on whether or not they will take testosterone or other kinds of anabolic steroids to improve their muscle power, and then ask them to sign up to give blood samples to see how their immune systems are responding to that. That kind of thing is particularly where you're dealing with hormonal manipulations and you have prior consent of well-informed people. I think it's possible to do ethical things. But if you want to really get into mechanistic detail, I think it's mice and rats. And you try to then use conclusions that you've drawn from mice and rats that are done in really invasive circumstances to try to generate stuff that you can then cross-check with more public health level, population level interventions on humans that are ethical. I can't see a way of actually directly addressing some of those things in humans in an ethical fashion.
Thank you. Rose, I hope that answers your question. Yasir says, is there a paradox in the idea that calorie restriction leads to longevity, autophagy, and lower inflammation rather than dormancy?
The reason I'm pausing is that there's a lot of complicated interactions in there. And so I'm trying to figure out whether it's actually a paradox or not. And I think that one of the reasons that it looks like that is that there's a real sort of dose response curve, and the amount of calorie restriction that you have to do to prolong the lives of organisms is really quite considerable. In humans, it starts to be effective when you get down to 800 calories a day, which is something I wouldn't want to do. Most of the information we have is from flies, mice, and worms. So I think I'm just going to have to back off and say, I don't really know that it's a paradox at this point. And I would have to sit down with a bunch of, the ideal situation for looking at is take 4 or 5 papers and take a bunch of grad students and sit down and chew it out around a table. And maybe you can come to a conclusion as to whether it's a paradox or not. I do think there are important interactions there.
Brilliant, thank you. Our next question, what are current thoughts on the presence of fetal cells in the mother mosaic? Is it an advantage enough to be the mother in terms of regenerative capacity, thereby dictating maternal mechanism?
That comes in from the senior author of the paper that I discussed.
I did, yeah. It's just a YouTube name. I'm not 100% sure. I was like, is that really? Hi, thanks for joining us.
Great paper. Well, the guy that really has thought a lot about that is David Haig. And so just to refresh everybody else's memory, any woman who has had a son has some cells in her body that have Y chromosomes in them. So there are cells that come into the maternal bloodstream from the fetus during pregnancy, and that creates mosaicism in the mother. And David has thought about the possibility that some of those cells would actually go in and target the ovary and try to, say, manipulate future reproduction of the mother based on their own genetic self-interest, because they would be dealing with the fates of future siblings. And they could their interests might be either to increase or decrease investment in them, or perhaps even intervene genetically in them. Those are wild and crazy ideas, and they're wonderful. And I don't really know whether there's any good evidence for it. It is one of those situations where you really want to be dealing with mice and rats, uh, not so much with humans. However, let me go on about that just for a second. I have wondered about the phenomenon of people who are transsexual and who feel that they are one gender, but that their body is another, so that their mind, their self-image or their emotional image of their own gender is not the one that their body has. And I have wondered about the possibility that that could be due to hormonal imprinting during pregnancy, and that some of those people had had a twin of the opposite sex who had disappeared during pregnancy. And we know that that actually is the fate of about 70% of conceptions that start as twins. About 70% of them end up being singletons. So many people out there who started life having a twin but didn't know it because that twin was resorbed before birth. And if it were of the opposite sex, it might have imprinted their brains at the time that their brains were developing that critical capacity to recognize what gender they were. Now, how to test that? One of the ways you could test it is with mosaicism. So, do transsexuals actually have any higher concentration of cells of the opposite sex in their bodies than do other people? One can imagine controls and treatments and things like that. However, when I investigated that, and I went down and I talked to people who were in human genetics at the Yale Medical School about whether this was a practical research program, they said, it's an interesting idea, but the concentration of such cells in the body is so low that you're probably below the detection level. And it's going to be really hard to get any evidence on it. At any rate, that's what I have thought about what mosaicism might tell us. Thank you.
Jerry Zhu, hi Professor, wonderful talk as usual. It seems that allergies are at least partly caused by mismatch. Do you have any thoughts as to why certain foods, i.e. shellfish or nuts, are more common as allergens than others? The second part of the question is cut off.
Yeah, hi Jerry, good to see you again. I don't think that there is anything systematic about that. I think that those allergens, we share a lot of the molecules that we find on the exterior of cells with a lot of other eukaryotic organisms. And I think that evolution has produced a lot of variation on that. And just happens that sometimes you get particular other molecules on the surface of cells of other species to which our immune cells react in a particular way. I think it's one of those large number things where if there are enough events, the improbable becomes probable. So that's not very helpful mechanistically. I have another student, Pascal Gagnon, who's now a professor at San Diego, who works a lot on the cell surface molecules. And it may very well be that there's something going on here that's epigenetic or has to do with early life experiences. But frankly, I don't know the mechanistic details. But Jerry, as you well know, my response would be, if you were a student in my class, would be, what a great paper topic. I think that you ought to go look up the literature. I don't know.
I hope that answers your question. We've got a few people saying, thank you very much for an interesting talk. As we wait for more questions to come in, I was really interested. I mean, I've read your book and it's in Oxford. It's not here, so I can't check because it's not online, unfortunately. But I was really interested in the graph about the different types of placentas and their evolutionary origin, because there's a similar-ish graph in Amira's 2012 paper about the evolution of menstruation. And I would have expected the two to really match up very well, but they don't, because Amira's hypothesis is that invasive placentas is what led to the evolution of menstruation exactly for this trade-off. I guess it's more kind of, I just really like to see the two graphs put together to see the differences between the two evolutionary origins in both invasive placentation and the evolution of menstruation. And I guess also kind of investigating if we've established that primates have higher rates of cancer than cows and horses, then maybe the other menstruating species like the spiny mouse and elephant shrew might do as well.
I think that's a very good point, and I understand from Dina's work on menstruation why you would be putting those two together. Now, Dina told me that she would be listening in at this point, and I think that she's probably sitting down there in the Buck Institute in California looking at us talk to each other. And I think that, Dina, if you could get together with Gabriella, that would be great. I think that you're in a much better position to make that connection than I am. Gabriella, I am currently actually editing a book that Dina is writing. And so, and I recommend that you have a look when it comes out. It's probably not going to be published until either later this year or early next year. She goes through a lot of these things. It's for a popular audience. She's trying to reach a broad audience with the book. But I think for a more technical response, it would be best if you and Dina simply got in touch with each other, either by email or Zoom, and traded thoughts on that issue.
Yeah, that would be great. If you're listening, I'll definitely reach out to you. Well, whether you're listening now or whether you're listening later on YouTube. So unless anybody else has any questions, if you do, please do drop them in the chat box now. We'll probably call it a day. Oh, Amira, yeah, hi, great. I would love to connect. That would be great. She's listening. Thank you.
Hi, Dina.
So thank you so much, Steve. It was really great to have you with us again online rather than in person, but that's the beauty of Zoom and YouTube. It means that we can connect even when we're on different continents. Next week, we will be hearing from Bernard Crespi on the 23rd of February at 4 p.m. And he'll be talking about the evolutionary biology of risks for endometriosis and PCOS, from fetal programming to life history. So I'm sure we'll be drawing on a lot of your work, Steve, and a very good continuation from your talk for next week's talk as well.
Yeah, I've read Bernie's paper. It's good stuff, so show up.
Well, there we go, endorsed by Steve Stearns. So come along next week. And thank you again for joining us. I'm going to stop the broadcast. So thank you everyone for watching.